Is it on? We're going to start in a couple minutes. We're just trying to get everything set up.
Where's the? Just turn the screen on.
Huh?
Turn the screen on. Hello? Hello? Okay, hello. Hello, everyone. Welcome to BIMM 101, BIMM 100 review session. We're going to go over the course material that has been covered so far. So I'll go over the basic structure of the DNA and like the first portion of the class, and then my colleagues here will go over DNA replication and DNA repair mechanism, and we will end with cloning and sequencing. Just one quick thing. Do not let this review be your only source of studying. Just make sure you go over the slides, lecture slides, podcast, all tests, and everything, because we were just covering the big picture, and we haven't seen the test yet, and we don't know what's on it. So we might be presenting something that is missing, and we did not cover it in lecture. Okay, so just make sure you go back to lecture slides and make sure you know everything in there. What else? Any questions, just in general, or no? Okay, so this is the central dogma that we talked about it, and this will be the topic of the class for the rest of the quarter. We start with the DNA, which is in our genome, like right in all cells, we have identical sequences of DNA. And to go to RNA, we use RNA polymerase and this whole process called transcription, right? To go back from RNA to DNA, we use another enzyme called reverse transcriptase, right? And you guys saw that in telomeres and telomerase, which we'll talk about later. RNA to protein, it's called translation, and we use ribosome and rRNA, which is also part of ribosomes, to get to proteins, right? And protein is a gene product, like many other gene products, right? The rRNA, tRNAs, and exist, which Dr. Ruther talked about in a class. For DNA to copy itself, it's called replication, and we use DNA polymerase. So this is just a general overview of what we're going to be talking about. Okay. Genes in our cells are made of DNA, which is in turns is made of sequences, and each sequence is made of a base, a nitrogen base, a sugar, and a phosphate, right? This structure alone, the base and the sugar, is called a nucleoside. When you add a phosphate to it, it's called a nucleotide, right? In DNA sequences, we use DNTP, which is nucleoside triphosphate. So we have a base, we have a sugar, and 3 phosphates of these. The D stands for deoxy, which is missing an OH and carbon 3. In this whole structure, the main components we'll be focused on is this 5' sugar and this 3' this prime carbon and this 3' this 5' carbon and this 3' the 3' carbon and the 5' carbon. The 5' carbon is attached to phosphate and always be attached to a phosphate, and the 3' carbon is attached to an OH, and these two are important for the polymerization when nucleotides add to elongate the strand. All right, any questions about this general structure? Okay. So the DNA structure is 2 strands antiparallel, right? One of them goes from 5' to 3' end, and the other will go from 5' to 3' end. And the bases are connected via a phosphodiester bond, right? And it's between the 3' carbon and 5' carbon that we just talked about it. So if I to circle a nucleotide here, I would circle sugar, base, and a phosphate that is attached to the 5' carbon. So it'd be like this. Right? Do not make the mistake and circle this, because then when I'm circling this area, the phosphate is attached to the 3' carbon, right? Okay, and it's a helical structure, and there are hydrogen bonds between nitrogen bases, right? And this portion is called the phosphate backbone. All right, this is just general. And any questions about this? Are you good? Okay, so as I said, when the strand is being elongated, we're adding the incoming nucleotide to the 3' carbon. So the OH will attack the phosphate of the incoming nucleotide and will result in these two phosphates leaving, and that releases energy. And you will create the phosphodiester bond here. Right, and we'll just continue going. Okay. The bases, there are two mnemonic to remember which base belong to which category. There's the cut pi and pure as gold. So as you can see, you have the C, U, and T, cytosine, uracil, and thymine with pyrimidines, right? And then you have, in purines, you have the A and G, right? Adenine and guanine. And another mnemonic to remind you how they base pair with each other, you have all tigers can roll. So you have the A pair, base pairs with T and C base pairs with G. Just to remember it real quick on test. You don't have to think about it and just do it. As I said, there are 3 hydrogen bonds between C and G and 2 hydrogen bonds between T and A. And so because we have more hydrogen bonds between C and G, that requires more heat to break these bonds during any reaction, whether it's PCR or transcription or any procedure that requires the breakage of these bonds. As you can see that pyrimidine have only 1 ring and purines have 2 rings, right? It's very simple. We have Chargaff rules, which is associated with bases, and it says that you have the number of the A's equals numbers of T's, and numbers of C's are equals to number of G's, right? But the combination of A and T is not equal to C and G, right? Pretty simple, not so hard. Any questions on this? We good? So one other thing about hydrogen bonds, 2 hydrogen bonds between 2 atoms, you have to have an H that is attached to a nitrogen or an oxygen or a fluorine, right? And that H should be also attached to another oxygen, fluorine, or nitrogen, right? Okay. Now about the ribose and the nucleotides. As you know by now, we have 2 ribose structure, which is just the regular ribose or the 2-deoxyribose. And this is what constitutes the difference between RNAs and DNAs. In RNAs, we are missing this OH in carbon number 2, but that is still present in RNA, right? So that's the difference between them. And RNA uses uracil, which is not used in DNA, right? It's just a general review. So I have a questions for you here. This is how you might be asked about Chargaff's rule. So if we have a combination of A and T that is 32%, find the percentage of the other 4 bases, right? So I'll just give you guys like a minute just to think about it real quick, and we'll go over it. What is the percentage of C plus G? Okay, what about A? T? C? And G, 34 is all right. So this was pretty simple, but it just demonstrates Chargaff's rule and shows the composition of bases. Any questions about this? Is it simple? We good? Cool? All right. So the last part of my presentation is about the discoveries that were made in the genome field in genetics and MolBio, and I'm going to start with Griffith, who demonstrated that genetic material is transferable, right? And he did that with the different strains of pneumonia when he injected mice with different strains, right? The smooth versus rough strain, and heat killing it, and then just seeing if the mice will survive or not. So this was the conclusion of his experiment. These guys, they determined that the genetic material is DNA, and they did that with the phenol and UV RNase and DNase, and see which strain is still infectious, or if the mice will die or like survive, right? And as you guys know, phenol is to disrupt protein structure, and UV disrupts DNA, and RNase disrupts RNA, right? Okay, and then we had Hershey and Chase. They did the work with the bacteriophage, where they incubated it with P32 and S35. And then they waited for that bacteriophage to infect a host cell and see which material was injected to that bacteria. And they found out it's DNA because the material that was injected was radio-labeled with P32, right? And as you guys know, P32 is only incorporated in nucleic acids, right? Franklin and Wilkins discovered the structure of DNA. They discovered that it's in helical structure, and they used X-ray. This is misspelled. It's crystal glad. Correct. You guys know what I mean. It's on there. We have Watson and Crick, and they also determined the DNA structure. And their work was based on Franklin's work. They used her pictures, but they expanded on it, and they talked about base pairing and how the strands are oriented regarding each other. Messelson and Stahl, they supported the semi-conservative model for replication. And I'm just going to go a little bit in detail about it. So before their experiment, there was three hypotheses proposed. There's the semi-conservative, and the conservative, and the disruptive, or dispersive. So for the semi-conservative model, the idea was that the two strands separate, and each of them will serve as a template for the new synthesized strand. For the conservative model, the two strands together serve as a template, and the new strand will be just synthesized out of that template, right? The dispersive model, you will have one strand containing the new and old strand. So it'd be dispersive, like this. So if we to take these three models and replicate them, this is what we will have. For the conservative model, you will have the parent cell, and this will not denature, or it could denature and come back and form the old strand again. But you will end up with the old strand and the new strand separate, right? They're not mixed. And so this is how they will continue. After a second round of replication, you will have the old strand still present with the other strands. Right? For the semi-conservative model, as I said, these two strands denature, and they will be part of the new synthesized strand. So they will have both incorporated. Right? And as you continue on the second round of replication, you will not see the old strand by itself, double-stranded. It will be divided within the new strands. And as you can see for the dispersive model, it's just mixed. You will not have a new versus old. It will be all just mixed like this. Right? Any questions about this? It's pretty simple, right? You good? So what they did is they incubated the bacteria with a heavy nitrogen, nitrogen 15, for a while, and then they incubated it with the light nitrogen, N14. And they just examined the structure after a couple rounds of replication to see which result they will get. And what they got was supporting this semi-conservative model. And I'll just show you how that was done. It's pretty cool to show you. So they used they centrifuged their solution, and this is what they got. So they started with the heavy, right? And after a couple rounds of replication, you can see that this is merging in the middle, and then it ends within the light side, within the light nitrogen, because that's, as you can see, this is what you will have at the end. You'll have the light, and you will have something that is in between the light and heavy. So at the end, they have some strand that only have the light nitrogen, and some strand that they have both, right? The light and heavy. All right. So this is my end of presentation. It was pretty simple. Are there any questions or comments or concerns? Yes. Yes. All right, any other questions? Yes. Which one? This one? So I don't have a picture, but I'll just try to draw it on the board. So they had a cell, pneumonia, which is a bacteria, right? And they treated it with different treatments. They used UV light, and they used RNase, and they used DNase, and they used phenol. Right? So they treated it with all these treatment, and then they injected it in the mice, and then they see if it survives or it doesn't, right? So just to see what is causing the genetic material to be infectious, right? Because I think they used a heat-killed pneumonia, and which is the smooth strain, and they mix it with the rough strain, which is not the infectious strain, right? And they just wanted to see which component of the smooth strain will cause the infection, and if it will transform to the rough strain, right? And so they used phenol, which denatures proteins, but it was still infectious, right? And then they used RNase, which denatures the mRNA, but it was still infectious. And then they used DNase and UV light, which denatures DNA, No, not together, separately. The different treatments, but they have the same effect. And so they did that, and they added it to their solution, and that denatures DNA. And when they injected that to the mice, they survived. The strain was not infectious anymore. Is there any other questions? Yes. Yes. I would say yes. Looking at his old tests, he does say who's like this and this, did this and this. So, yes. Yeah. 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 You will have strands of mixed isotopes, single strand of mixed isotopes. So you see this single strand by itself? It has light and heavy. Yes, yes, you're right. So there's no difference between this and this. They're both intermediates. It's just how they're represented. Yeah, definitely right. Okay, any other questions? Um, if not, I'll turn it into Karthik.
Can you guys hear me? All right, guys. So, yeah, I have a little bit of overlap with Rand's slides. I just want to talk about strand polymerization again. So the main idea here is that your 3' hydroxyl group is going to attack. So hydroxyl groups are good nucleophiles, right? So the hydroxyl group will attack this phosphate bond right here, and it'll form a phosphodiester bond, and that's going to create the sugar phosphate backbone. So what would happen, just a question to you guys, what would happen if you didn't have this 3' hydroxyl group? Anyone? Yeah, so if this nucleotide sits down and you add more nucleotides, if there's no 3' hydroxyl group, it's not going to be able to create the next phosphodiester bond. So that's what you see with Sanger sequencing and dideoxynucleotides. Okay, so lagging strand, or the leading strand synthesis. So the best way to recognize which one is your lagging strand and which one's your leading strand is to see the direction of replication for that strand. So if you're going into the fork, that's going to be your leading strand, right? So it's always 5 to 3. Okay, so if professor just gives you a picture like this, you should be able to tell which one's going to be the leading and which one's going to be the lagging, right? So you have to remember that the replication is bidirectional, so you're going to have a leading and lagging strand on both sides of the replication fork, right? So let's just look at this side first. So which one would be the leading and which one would be the lagging? Anyone? Yeah, so the top would be the leading, right? So this would be your leading, because if you were going to replicate, you'd go in this direction from 5 to 3 prime, right? So that would make this strand your lagging strand. Okay, so what about on this side? of the replication fork. So it would be the opposite, right? So you would go in this direction for the bottom strand. So this would make it your leading strand. And then this would be your lagging strand. So the easiest way to look at this is your leading strand is going to be replicating into the fork. So that's always going to be your leading strand on both sides. Yeah. Where? Oh, sorry. Yeah, you're right. Yeah. But yeah, it's still a leading strand on bottom. Okay, so right. Okay, so let's just talk about leading strand synthesis first. So I'm just going to draw one side of the replication fork. Okay, yeah, I forgot to mention, if you guys have any questions and you don't want to ask the whole class, please feel free to come down and ask one of the TAs. Okay, so what would be the first step of leading strand synthesis? Yeah, so helicase is going to start unwinding the DNA. And that requires ATP. Why does it require ATP? Yeah, so it takes energy to tear apart the hydrogen bonds between the two strands, right? Okay. So what would be the next step? Yeah? Even before that. So what does polymerase need to start replication? A primer, right? So you have a primase. So the primase is going to start putting down an RNA primer here, right? And this would be in the 5 to 3 prime direction. Okay, so now what's the next step? Yeah, so you have your DNA polymerase. And it's going to start extending in this direction. And what's the last piece of the puzzle? So if you're unwinding the DNA, it's all wound up here. If you're unwinding the DNA, you're going to create a lot of tension over here, right? So what prevents the tension from stopping the polymerase? Yeah, so you have your topoisomerase. Okay. All right, so any questions on leading strand? Topoisomerase, sorry. And that also requires ATP. Yeah. No, it doesn't. Yeah. And why doesn't it require ATP? Why doesn't DNA polymerase require ATP? Exactly, yeah. So the reaction itself provides enough energy. When you break the phosphate bond, it creates enough energy for the reaction to progress. So any questions on leading strand? Yeah. Okay, so this is lagging strand synthesis. Yeah. So sorry about that. So once you get to the end of this fork, or once you've got to the end of the region that this polymerase is replicating, while it's replicating, an RNase will come in and remove this primer and fill it in. Okay, so the best way to recognize which one is your lagging strand is to see which strand is replicating away from the fork. So the leading is always going into the fork, and the lagging is going away. Okay, so what would be your first step on lagging strand synthesis? What was that? Yeah, so helicase again. Okay, and the next step. Yeah, so you have your primer here, and it's going to extend away from the fork, right? So if you were to move forward in time, so you have this primer right here, which has extended forward, and your helicase is opening up the strand, right? So now you're going to have another primer that sits down here, made by primase, and it's going to start extending this way. And while this primer is being extended, RNase H will come in and remove one of the primers before, and DNA polymerase will come in and start to fill in the gap right here. Any questions on this? And once DNA polymerase gets to the end of the next strand, what's the last step that has to happen to join the two strands? Yeah, so ligase will fill in the phosphodiester bond. Yeah. I don't think it's important. Yeah, so one of them is important. One of them is just for regular DNA extension, and the other one is for filling in gaps once the primer has been removed. But I don't even know the difference. I don't know which one's which. Okay, so let's say that you've replicated both strands of DNA. So this one would be your leading. And this would be your lagging. So this was your last primer, which was made, and it's at the 5' end. So which one is in the lagging strand, which one's the parental strand, and which one is the newly synthesized strand? Yeah, so the top one is the parental, right? Okay, and the bottom one would be your daughter. And you can tell that because the daughter strand has the last primer, right? So this is your last primer, so RNase H is going to come in and wipe out the last primer, right? So the next step you'd think would be DNA polymerase would fill the gap in, right? But DNA polymerase needs a 3' end of DNA to sit down and start extending. So since there's no 3' end of DNA on this side of the strand, this would present a problem because DNA would shorten, right? So if you have open ends of DNA, nucleases come in and start breaking down the DNA. So the solution that your cells have is telomerase. Okay. Yeah, just to reiterate, so if you did not have telomerase, you would have this open end of DNA. Okay, so which strand does telomerase elongate? So this is a quiz question. I think a lot of people missed it. So which strand does it elongate? So it's the lagging strand, and then which lagging strand? So within your lagging strand, you have a daughter and a parental strand. So a daughter and a template, right? So which one does it elongate? The parental strand, right? So in this picture, I guess this is backwards from what I drew. But this would be your 3' parental strand. And you know that because here the primer has been removed on the daughter strand. So only the daughter strand will have primers, right? So once it's been removed, you have this, this open gap right here. And this is where telomerase will come in and elongate. But it's going to elongate the parental strand. So bases will always be added to the 3' end of the parental strand. Okay, so what are the components of telomerase? So there's two of them. Yeah, so you have an RNA template and you have your reverse transcriptase. So this is these are two components, but they're in one enzyme, right? So they're both in one package. Right. So telomerase is going to recognize a repeat at the end of the DNA. It's going to sit down. So this AACCCC recognizes TTGGGG on the parental 3' end. And once it recognizes this, it's going to start adding nucleotides. So it's going to add a TTG. And once it's added a TTG, it creates a recognition site for itself. So it's going to be able to translocate and bind to the DNA again. So it's going to lift off. It's going to lift off. And since it created this TTG, it already knows where to sit down and bind again. So it's going to lift off and then bind on the TTG. And it's going to keep creating repeated base pairs. And the base pairs themselves will have the binding site for the next repeat. Does this make sense? Does this make sense? Any questions on this? Okay. So the ends are protected by, so once you have this end created, it's going to be repeated for hundreds of times. And the reason that telomerase works is because the ends will be protected by telomeric proteins and also by looping of the DNA, so that the nuclease will not be able to chop up the end of the DNA. Any questions on this? All right. So any other general questions? I'm going to move on to the problems. Yeah. Oh, why it's, yeah, so why is it upregulated in cancer? So the reason is, so cancer cells are dividing much faster than your normal cells, right? They're uncontrolled growth. So if you did not have upregulated telomeres, then since they're multiplying so fast, you would start chewing up the end of the DNA because it would start shortening. So they upregulate the telomerase enzymes so that they have really, really long telomerase, really, really, really long telomere repeats at the ends of the DNA. Yeah, so in cancer, they're dividing really fast, and they're dividing a lot. So if you didn't have long telomeric repeats at the ends of the DNA, then the DNA would just keep shortening. If you had the telomerase enzyme working at the normal speed that it normally works in your regular cells. So since cancer cells are replicating so quickly, they need upregulated telomerase so that they can maintain the really long telomeric repeats. Any other questions? No? Okay. So I have a couple of multiple choice questions from the practice test, and then I made some short answer questions as well. So the first question is, which cluster of terms accurately reflects the nature of DNA replication in prokaryotes? Give you a couple seconds. Does anyone have an answer? Yeah, so the answer is E, right? So in your eukaryotic cells, you have lots and lots of origins of replications because the DNA is so long. If you just had one, it would take forever to replicate all your DNA, right? So it's going to have multiple origins of replication throughout the DNA, and it's going to replicate in both directions. Whereas prokaryotes generally only have one origin of replication, and it's still bidirectional. And then semi-conservative. Okay, so number 2. Are any questions about number 1? No? Okay, so number 2, in DNA, phosphate groups bond to. Does anyone have the answer? Yeah, C, right? So your 5' phosphate group will be attacked by the 3' hydroxyl group of the deoxyribose from the previous nucleotide. Does that make sense? Okay, number 3, how are telomeres protected from enzymatic degradation? Okay, this is an easy one. Yeah, D. Yeah, so you have the longer repeats at the end, and proteins are going to sit down and bind on the DNA, and they're going to block nucleases from chopping up the DNA. Okay, and the last one, telomerase is composed of, yeah, so RNA and reverse transcriptase. Okay, so I drew this picture on paint, so if it's like really unclear, I'm sorry about that. So the first question, so you have this replication bubble right here, and a couple questions. So where are the 3' and 5' ends of the DNA? So just by looking at this, you should be able to see where the 3' and 5' ends are based on where the primers are and where the strands are. So where would the 3' and 5' ends be? So let's just look at the left side. So which one would be the 5 and which one would be the 3? Yeah, so 5' would be on the top and 3' would be on the bottom. And why is that? Yeah, so this is your leading strand, and you know it's going into the fork, right? So if the newly synthesized strand is 5 to 3, then this must be 5 to 3. Okay. So where would the leading and the lagging strand be? So I just said that before. So the top strand would be the leading, and the bottom would be the lagging. Okay, so where are the DNA polymerases in this diagram? So there's 6 of them. So for sure, you're going to have one here and here at the ends of the DNA, right? And there's 4 more. Anyone? Yeah, so they're going to be near where the primers are being elongated, right? So these 2 primers right here are being elongated, and so are these. But if you can see, the primer here is shorter, so that means the primer is still being synthesized. So you wouldn't have DNA polymerase at these 2 primers. You'd only have them at the inside 2 primers. Okay, which primers were synthesized first, second, and third? So let's look at these 3. So which one would be the first one? Yeah, so the one closer to the middle of the bubble, right? And then as you opened up the replication fork, then the following primers would be synthesized. Does that make sense? Yeah. Okay, and the last one is, where is primase currently at? Yeah. Yeah, so it's going to be at the ends, right? So it's synthesizing these 2 primers right here. So that's where primase would be at. Any questions about this? No? Okay. So the last one. So in this problem, we are exploring telomerase, the telomerase enzyme in vitro, right? So this is in a test tube. So what you did here is you have this DNA template, and you put the DNA template and the telomerase enzyme into a test tube, and you're going to be giving it different conditions to elongate, right? So the first question is, label the 5' and 3' ends of the telomere RNA and DNA template. So what would this end right here be? So the easier way to think about this is, what would this end of the DNA be? Would it be 5' or 3'? It would be 3', right? So if this end of the DNA is 3', that means this end of the RNA must be 5', right? So your DNA and RNA are always anti-parallel. Okay. What telomeric repeat would be produced if you gave the reaction all the DNTPs, right? So you're kind of doing PCR here, except with telomerase and your DNA, right? So if you gave it all DNTPs, what sequence would you produce? So what 3 base pairs would we produce here? Yeah, so its own state. So it would be TTG, right? So you have TTGGG, and then you'd produce another TTG, right? Okay. And then it would translocate and then keep producing repeats, right? Okay. Number 4, what sequence would you produce if you only provided the reaction DTTP? So what is DTTP? So it's just your thymine nucleotides, right? So if you only provided the reaction DTTP, what sequence would be produced here? So would you produce the Gs? No, right? So there's no DGTP in the reaction, right? So you just get TT, and then it would just stop. Does that make sense? Yeah? Okay. Number 4, what sequence would you produce if you change the last 2 repeats on the DNA to AACCCC, AACCCC? What sequence would you produce? kind of tricky. No? So you wouldn't produce anything. So why wouldn't you produce anything? Yeah, so if you don't have this sequence for this specific telomerase in your DNA, if you don't have this TTGGGG, your telomerase will not be able to bind. So even if you provided all the DNTPs, it's not going to be able to recognize and find the DNA sequence. So it will not produce any sequence at all. Does that make sense? Yeah? Okay. And the last one. What sequence is produced if you change the telomeric RNA sequence to this? So this right here, AACCCCGGG. So right here, AACCCC, or GGG. So you're changing these last three to GGG, or these three right here. So AAC becomes GGG. You'd add CCC, right? And then would you keep going after that? And why wouldn't it keep going? Yeah, exactly. So these base pairs right here are part of the recognition site for the telomerase to move over and sit down again, right? So if it produced a CCC right here, then this AAC will not be able to bind to the CCC. Does that make sense? Okay. So any other questions about DNA replication in telomerase? Yep. Okay. So I'm going to hand it off to Safwan.
Hello? All right. What's up, guys? All right, so I'll be covering DNA repair mechanisms and basic genome stuff and sequencing. All right, to start off with the basic outline for DNA repair, it's just from the slides, you recognize the damage or mutation in the strands. You excise out that damaged or mutated DNA strand, or portion of it, and you fill in the gap with DNA polymerase and ligase. So that's just the general outlook. All right, now, sorry this is a little clustered, but I'm going to walk you through it. You can have just proofreading activity by DNA polymerase, 3' to 5' exonuclease. You can have DNA single-strand repair, mismatch excision repair, or base excision repair, or nucleotide excision repair. And the mnemonic that Dr. Ruther used in class was Mr. Bill Clinton, naughty Democrat. All right. And then you can have DNA double-strand repair, which includes homologous recombination. That's usually error-free, or always error-free, and end joining. And that can have errors. Okay, single-strand repair. Mismatch excision repair basically looks for just erroneous DNA and removes it. 1 second. All right, yeah. So it recognizes the incorrect DNA on the daughter strand. Now the question is, why don't you have the parent strand being attacked by endonuclease activity? Why does it, recognize the daughter strand? Yeah, because the parent strand is methylated, so you cannot have any endonuclease activity on that. So the mutated DNA is removed by endonuclease, and to make things a little more smoother, exonuclease takes out a couple more bases, and after that, DNA polymerase and ligase fills in the gap. Pretty simple. All right, base excision repair. Base excision repair fixes chemically changed DNA. So it's a pretty simultaneous process that methylated cytosine changes to thymine, and that it happens by the process of deamination. So you guys should remember the process. The chemically changed base is then repaired. So they remove the T and they put back the C in there. So you recognize, or you remove the chemically changed base by glycosylase, and the nuclease removes the deoxyribose phosphate backbone, and then you fill in the gap with DNA polymerase and ligase. And just go back to the slides if you have problems visualizing it, but then just know the details and the sequence in which it is happening. Nucleotide excision repair. Now that fixes distorted DNA. So you know that UV light damages DNA. How? Can anyone tell me? What? Sorry? That's correct. So UV radiation makes thymine dimers. So it just changes two normal T's into a dimer T. So you recognize the distortion. You open the helix after that. Endonuclease removes the damaged dimer or DNA, and you fill in the gap with DNA polymerase and ligase. So I'm going to stress on the fact that just know the sequences for all these single-strand repair mechanisms. Just practice it a couple times. If I asked you a question what's special about base excision repair? How's it different from the other two single-strand repair mechanisms? What's special, one way it's special, the repair mechanism. Yeah, glycosylase only takes out the damaged nucleotide. And then you have endonuclease come in and break up the backbone. of the DNA strand. So that's how it's different. The other ones are just like endonuclease right away. But then over in base excision repair, you have glycosylase. All right. Double-stranded repair mechanisms. So you have like two of them. Homologous recombination. Like I said before, it's error-free because you're using the sister chromatid as a template to fix it. And I'll run you through the entire process here. So it's similar to crossover during prophase 1 of meiosis, but over here the exchange is happening between sister chromatids and not chromosomes from different parents. Okay? It's like if you picture it like it's the same parent with two chromosomes, and you're kind of I think she's drawing it out, and then I'm gonna go over it when she's done. But yeah, before I get to the process that Dr. Ruther covered in class, I'm just going to talk a little bit about holiday junctions. So just know that you disrupt the holiday junction by breaking the phosphodiester bonds in the backbone of the DNA. Now you can have a non-crossover product or a crossover product. So you get the non-crossover product by basically cleaving at the holiday junctions. All right, I'm going to draw that separately over here. One sec. All right, so as you can see over here, you have, you can have two holiday junctions over here. Now there's two ways to cleave this. You can cleave it right there and there. So cleaving it at the junctions will give you the non-crossover product. And in that case, you're going to get identical sequences with different origins. So after the repair mechanism is over, the fixed strands are going to be identical, but they'll have different sequences because they came from the two different sister chromatids. Can you guys see it on the board? Like, yes, no? Yeah, it's a non-crossover product if you cleave it, at the junctions of the holiday structures. Does that? It's a good question, Randino. Yeah, she's gonna answer that.
How you asking about the mechanism? Why is it not crossing over? We can draw it on the board if you want to know like how that happens. Just go back to Dr. Ruther's videos. It just shows you how when you break this, how these two will reconnect together, and the upper strand will not incorporate any of the other strand, so you will not have any crossing over. It's just hard to draw on the board. But the gist of it is if you break the phosphodiester bonds at the holiday junctions at the cross ends only, you will not have any crossing over with the outer strands. You will only have a heteroduplex within the, yeah, like you will not get a total crossing over. We will not have a swap. The outer strand will not play a role in this whole process if you're only going to cut in the holiday junctions, okay? And you want to continue?
Yeah, sorry about that. It's kind of like hard to picture for me as well. But then just look over the videos. That should help you a lot. But yeah, so this would be the non-crossover product. You can also have a cleave at, say like one holiday junction, and the other one you'd have a cleaving at, the parental, or the parental strands basically. So that way you can have a crossover product because both strands are changing. Does that make sense? So if you have one holiday junction cleave and the parental strand cleave, that's going to be a crossover product. Okay, and then, so there could be a total swap by breaking the bonds at the cross ends and the other strand as well, just like I explained. So the basic mechanism of homologous recombination, the one that Dr. Ruther covered, was the one with the crossover product. Which two enzymes? Question. I'm not clear as to what you're asking. Uh-huh. Well, so, I mean, I think during the crossover product phase, the sister chromatid will be used as a template to fix the broken strand. Yeah, I mean the one that crossed over. So that's the one you're going to be fixing. All right. One second. Okay, let me go over this one more time. If you have the non-crossover product, it's just basically between these two strands right here, like this strand right here and this strand right here. In that case, you won't have the sister chromatid being used that much, right? I mean, you don't use that for the repair. But if you have the complete crossover product, where you cleave the parental strand as well, in that case, you use the sister chromatid to fix it. Does that answer your question? Don't get bogged down by too much of the details for homologous recombination. If you have to take away a basic message, it's just that you use the sister chromatids as a template, right? And just go over the podcast and the videos. That should help you visualize it a little better. I think you need to worry about that, but then you're asking how these strands, yeah. Well, I mean, the portions that you switch, those are identical on both strands. They're just different origins. So, like I said, going back. Yeah, these strands were identical. They're in the same direction. So when you're swapping, you still, conserve the direction. Yeah, if you want to imagine that way, or sense, antisense, antisense, sense. Does that make sense? All right. Okay, sorry if I did a horrible job explaining that, but then, yeah, don't worry about too much of the details. But yeah, I was asking you guys which two enzymes are responsible for the strand invasion. That's something, you might be asked. What are the enzymes that, allow strand invasion for homologous recombination? What two enzymes catalyze strand invasion for homologous recombination? There's RAD51 and REC-A. Do you guys remember those two enzymes? So yeah, when you're going over the process step by step, those two enzymes you definitely have to know, because he might ask you a question. RAD51 in eukaryotes allow for strand invasion during homologous recombination. Okay. All right. I'm sorry? So RAD51 in eukaryotes and REC-A in prokaryotes. Okay? All right. So end joining. Basic sequence, the broken ends are recognized. You have an exonuclease activity to remove the overhanging nuts, so that both are nice and symmetrical, and then you ligate the two ends. So when this process happens, sometimes there are errors. The leading example that Dr. Ruther gave you in class was translocation between chromosome 9 and 22, and you get the result is the Philadelphia 22 chromosome. So it's not a regular 22 chromosome, but it has portions of chromosome 9 in it. And this messed up chromosome can lead to leukemia. So definitely know that example. And from the table of possible diseases resulting from DNA repair defects, kind of go through, the diseases over there, but then I would ask you to know at least xeroderma pigmentosum, which is a form of skin cancer that happens from nucleotide excision repair defect. And the other one you'd probably want to know is hereditary breast cancer. So when you have deficiencies in the BRCA1 and BRCA2 genes. So in that case, you have defect in homologous recombination for the breast cancer. All right, make sense? So genome, what's in a genome? You have single copy genes, you have introns, you have gene families, spacer DNA, simple sequence DNA, mobile DNA elements. So just kind of have a basic idea, all right, what's in a genome. And then have an approximate idea of genome sizes. So if I ask you, if you have a bigger genome, do you have more genes? No, you don't. And an example would be between mice and humans. So humans have a bigger genome, but they have less genes than mice, which have smaller genome. Okay. And definitely know the biggest genome belongs to flowering plants because of polyploidy. And then, so from the Human Genome Project, you want to know like these stats that the human genome has 3.1 billion base pairs. The variation between human genomes happens due to like SNPs and CNVs, so single nucleotide polymorphisms and copy number variations. And then 50% of the human genome comes from transposable elements and other repetitive DNA. Okay? All right, DNA synthesis. So what are the 4 things that you need for general DNA synthesis? Can someone tell me? What's the first thing you need for DNA synthesis? What are you going to copy? A DNA template. What do you need for the new DNA strand? What needs to come in? DNTPs. You need primers to start the process as well, and you need DNA polymerase, the enzyme that's going to actually allow the synthesis. PCR basically works on this concept to amplify DNA, and you can have it for multiple reasons. The process of PCR, first you heat up to 95 degrees Celsius. I would approximately know the temperatures for PCR. So initially you heat up to 95 degrees Celsius. That's gonna break up the 2 DNA strands or denature it. Then you cool down to 50 degrees Celsius, and that's gonna anneal the primers. And then you heat it up again to 72 degrees Celsius, and at that temperature you have optimal DNA polymerase activity. So what kind of DNA polymerase are we using here? Yeah, it's like why is it so stable at high temperatures? Because you're taking it from a heat-stable bacteria, Firmus aquaticus. Again, something you should know, Taq polymerase. All right, Sanger sequencing. It works on the same idea as PCR, but instead of just regular DNTPs, Sanger sequencing also uses ddNTPs. Now, ddNTPs are special because you don't have the 3' hydroxyl group, which you have in DNTPs. So if you incorporate ddNTP into the chain, you'll have no more elongation. So in Sanger sequencing, you can fluorescently mark these ddNTPs to see where the sequence ends, and that's how you can sequence the whole piece of DNA that you're trying to identify. All right. So for Sanger sequencing, you have to move the fragments through agarose or polyacrylamide gel with an electric field. Remember that short sequences will be towards the positive electrode, and as you go longer and longer, you'll move towards the negative electrode. So which electrode will, the 5' end of DNA strands, or the sequences will be? Like which end will it go to? So the 5' end of DNA, which electrode will that proceed towards? The positive or negative? Positive. So know that DNA elongation happens from 5' to 3'. So the 5' end will go towards the positive electrode. All right. Another type of sequencing that we learned about was SBS, sequencing by synthesis. Over here, basically, you have beads that are complementary to an oligo, which is a sequence of nucleotides that are synthesized. The oligos are going to basically be primers. They're going to be on the ends of the sequence that you want to identify. You're going to use fluorescent nucleotides to anneal to your sequence of interest. So it's basically one fluorescent nucleotide comes in, you take a picture of that, and then you wash it away. And then another fluorescent nucleotide comes in, so forth, so forth. So from the pictures, you can kind of decipher what the sequence was by complementary base pairing. Does that make sense? So yeah, for sure. So suppose you have oligo here, and this is your sequence of interest. So this part of the oligo will, bind to the beads in the well. And then, okay, the fluorescent nucleotide comes in here, binds. You take a picture, and then you wash away the nucleotide. And then, again, you repeat the process. Another fluorescent nucleotide comes in, binds to the second base. I'm sorry? It washes away the full nucleotide. All right? So basically, from the pictures, what the sequence was because you took a picture of the fluorescence, right? And that will tell you what base it was. So from that, you build up the whole sequence from the pictures, and you decipher, okay, what was the original strand it was binding to. I'm not sure about that, but then just know that you just go one nucleotide at a time. That's all you need to really worry about. Yeah, you wash away the nucleotide, you put in a new one, you wash away that, and so forth, so forth. And just keep repeating until you have like a picture of all the. So you're not actually creating a strand? No. No. It's going to be one at a time, right? Like this. What? You just wash away the fluorescence? All right, I'm sorry, guys. You basically wash away the fluorescence, all right? But yeah, the sequence, the nucleotides that came in, they remain. And yeah, the strand is created, but then from the pictures, you'll decipher what the nucleotides were from the fluorescence, and you can work backwards to know what the original sequence that we were sequencing was. Make sense? All right. All right. After that, you can have the nanopore sequencing. It just basically uses a protein pore. You pass the DNA sequence through that, and then through disturbances in the current, or through characteristic disruption, you know what the DNA sequence was. Okay? All right. Whole genome sequencing. So know the three-step approach. Linkage mapping. That's going to give you the order of genetic markers. Then you have physical mapping. You have the ordering of large overlapping fragments and short overlapping fragments. And then from the overlapping fragments, you build up the DNA sequence. And you determine, what the sequence was. All right, shotgun approach. The computer does, all the work for you. So you have an unknown genome of interest. You copy that genome multiple times. You break it up. You break up those cloned genomes into fragments, and the computer basically aligns those random nucleotides and gives you or constructs the whole genome and tells you what the genome was. Does that make sense? Okay, proceeding on. All right, so we already went over, what's in a genome, but I'm going to focus on single copy genes and gene families. So what's a gene? Can anyone tell me what's a gene? Anyone? Okay. A gene is a DNA sequence that makes gene product. So it can be either coding or non-coding RNAs, or it could be proteins. So the primary product is always going to be RNA, whether it's mRNA, which is coding RNA, or some other form of RNA, which is non-coding. In terms of genes, they can be solitary genes, they can be by themselves, or they can be duplicated genes. So you can have many copies of the same gene. Now, if you have duplicated genes, they can be similar to each other or exact copies. Example of exact copies would be the rRNA gene. You need a lot of rRNA to make up ribosomes. So you'll have a lot of copies of the rRNA gene, and they're exactly same, in terms of sequence. Or you can have the human beta globin gene. The copies might be nearly identical, or similar, or non-functional. The non-functional genes, those are what we call pseudogenes. So in terms of sequence, they're similar to their functional counterpart, but they don't produce anything anymore. All right, last, how do you know which parts of DNA codes for RNA? So this is where the cDNA library comes in. So you take RNA from the cell. By hybridization methods, you make a cDNA library, and then you sequence it to see what the nucleotide sequence was, or you bind it to the DNA to see where it binds to in the genome, and that way you can find out the location of the genes that code for mRNA. Okay. Try a couple of practice questions. How do you All right, so can someone answer the first question? What is a gene? Just so I went over it. Anyone? Right, thank you. So a gene is a DNA sequence that codes for gene product. Now, gene products can be two things. What can be possible gene products that we went over? Proteins, another one. RNA. So it can be proteins or RNA as gene products. All right, true or false? The promoter region is not considered part of a gene. I don't think we covered that yet, so all right, never mind about that. Okay. True or false? The majority of our genome codes for gene products with known function. False. I think only about 1.5% of the total genome codes for gene products that actually matter. Yeah, and definitely know that number. 1.5% produces gene products. During mRNA processing, introns or exons are removed from the pre-RNA. We didn't cover that, did we? No, okay. All right, never mind about that question either. All right, back online. Okay, did you say this RNA? Yeah, that's correct. So some more examples would be mRNA, tRNA, and telomerase RNA. So these are the examples that Dr. Ruther wanted you to remember. So definitely know those. So which types of DNA sequence are the basis of DNA fingerprinting? All right, we're going to cover that in a little bit, so don't worry about it right now. Okay, so what's cDNA? Can one person tell me? Yeah, it's complementary DNA. So by hybridization methods, you bind DNA nucleotides to RNA to find out which parts of the DNA actually coded for RNA. So how does it differ from genomic DNA? Can anyone tell me that? All right, so in the genomic DNA, you can also have spacer DNA or non-coding DNA, DNA that doesn't code for any gene products. So that's how cDNA is different from the normal genomic DNA. All right, so going on to PCR. Why is it necessary to add 2 primers to carry out a PCR? And we have a hint up there. Like, what would happen if you just had 1 primer instead of 2? No, actually, if you had one primer instead of 2, you'd get linear amplification instead of exponential amplification. So because you have 2 primers, after 30 cycles, you're left with 2 to the power 30 copies of the DNA of interest. If you had just 1 primer, you'd just be left with like 30 copies. All right, so 14B, what would the product be and how much product would you get if only 1 primer was added? Just like I said, like 30 copies instead of 2 to the power 30 copies. Also, it wouldn't be Um, yeah. Yeah. So. Yeah. So whatever Josh said over there, you'd basically get just like a runoff from like the single strand instead of what was it? Like, sorry. Okay. Instead of specific segment, it would be like random products. Um, so why is Taq polymerase used and not a DNA polymerase? Because Taq polymerase is heat stable, and we need that for the high temperatures for PCR. All right, running through the process, what happens at 95 degrees Celsius for PCR? I just heard a jumbled answer. Can someone tell me? One person. What happens at 95 degrees Celsius? Yeah, DNA denatures. You separate out the two strands. Um, okay, what happens at 50 degrees Celsius? Yeah, the primers anneal, and that's going to start off the whole process for PCR. What happens at 72 degrees Celsius? Okay, I heard DNA polymerase. Yeah, so the DNA polymerase activity happens at this temperature because it's the optimum temperature for Taq DNA polymerase. All right. I'll post this problem set onto TED after the review session. So if you have confusions, just feel free to email me, okay? All right. Guess I'll be handing it over to Josh for
Having trouble, so take a minute to discuss and reflect. Oh,
Yeah, that's cool.
awesome. Sorry. Hello? Can everyone hear me okay? Thumbs up in the back? Cool. All right. So it's been an hour and 45 minutes. I'm sure everyone's getting antsy. So what I'm going to do is go through this last section kind of quickly, give you some of the main points, and then if you're feeling good we can like take a stretch break. Everyone can leave if they want to, or stick around, and I'll be here to go more in depth with anything that you're having trouble with, okay? So I don't have my own slides. I'm going to go through the lecture slides. And there's a lot of information on the lecture slides. Don't get overwhelmed by it. Just listen to what I'm telling you. I'm going to tell you what to kind of focus on, okay? Okay. So the next section is repeated segments of DNA. And so we have satellites and microsatellites. The name should be pretty self-explanatory. Microsatellites are smaller than satellites. Don't worry too much about the numbers here. You don't need to remember 14 to 500 or 1 to 13 base pairs. Just have an idea of kind of the order of magnitude, the relative order of magnitude. So with satellites, you're looking at 10 to 10 to the second power repeats, whereas microsatellites are going to be like single digits, maybe 10, on the order of 10 base pairs in a repeat. And then 20,000 to 100,000 base pairs versus like 100 base pairs. Just have an idea of the relative orders of magnitude there. Don't focus on the specific numbers. How do these things come about? So they can grow by recombination or crossover, where the repeats, because they're repeated sequences, during crossover, the machinery tries to line up similar or identical sequences. But when you have a bunch of repeated sequences in a row, the machinery, it's just a protein. It can't tell that this is repeat number 8 and this is repeat number 6. So it's just trying to line up sequences that seem similar. So when it does that, it can align repeats that are essentially the wrong order. So in this case, we're lining up repeat number 6 with repeat number 4, and that produces one longer satellite and one shorter satellite. And this is just another slide saying the same thing. You get n minus x, basically, and n plus x, where n is the number of repeats before the crossover, and x is basically the number of repeats that it misses by. So in this case, the machinery missed by one repeat, so you get one satellite that has n minus 1 and one satellite that has n plus 1. Is anyone having trouble with that, or is that pretty clear? Okay. Another way these can grow is backward slippage during DNA replication. So again, there's just all these sequences that do the exact same thing in a row. The DNA polymerase is going along and copying this. If some of the DNA way back here where it's already copied slips, it all slips backward, the DNA polymerase is still going along and just chugging along these repeats. So where this section of DNA slipped right here, that's going to become an additional repeat. And that increase in the length of the satellite will propagate and as the chromosome gets replicated generation after generation. And so that's another way that satellites can grow. PCR, unless anyone is really having trouble, I'm not going to go over this again. I feel like we've beaten PCR into the ground already. I'm also not going to go over gels unless anyone really has a specific question right now. Yeah? Sorry? Sorry, did you say on TED? So this is the lecture slides on TED that I'm going through. So this is all stuff that you already have access to. Not going to go over ladders. We can, if you want to, we can stick around and I'll answer your questions. Fingerprinting, again, stick around if you want more details. Okay, so now we're looking at the relative contributions of these different types of DNA to the genome. And you see here that protein-coding genes are a tiny, tiny portion of the genome. So what's all that other stuff doing in there? We have these, well, there's repetitious DNA that is covered, and there's these transposable elements, these mobile elements of DNA. So you're looking at this and you're seeing, hey, there's these pieces of DNA that move around, And for example, a transposon, all a transposon does is encode a protein that recognizes the transposon and moves it from one place to another. So a common question I get about this is, why? What does this do? What is its purpose? Can anyone answer that question? Why do transposons exist? Yeah? Huh? For their own benefit, yeah. So this is kind of an example of what you might call a selfish gene. Remember, nothing in biology makes sense except in the light of evolution. So how might have transposable elements been selected for? All they do is replicate themselves. And a gene, remember, a gene that can create more copies of itself will be more represented in the next generation and hence be selecting for itself. That's all a transposon does. It's basically like a virus. It's like a parasite. It only exists because it can copy itself and grow within the genome. So the question of what is its purpose, why is this here, kind of comes from a perspective of like design, right? Or that every single thing in a cell has to have a specific function that helps the organism survive. Not the case. This is basically a genomic parasite. So remember, there's two basic mechanisms of transposition. One is cut and paste, one is copy and paste. It's kind of like just Microsoft Office. So here's a transposon, and again, all the transposon does is encode for transposase. Transposase recognizes the transposon, cuts it out, and moves it somewhere else. That's all it does. You notice on the slide here, there's those little black sections that are labeled as direct repeats. Just a little additional information on that. The reason there are direct repeats on either side of the transposon has to do with the mechanism of cutting out. So what happens is the DNA gets looped, and these direct repeats line up with each other. And then the transposase can cut this out. So that you end up with the DNA where the transposon used to be and this circle of the gene for transposase. And then it can get moved somewhere else. Okay, this is, again, a lot of information on this slide. I'm not going to go through that all, but this shows you the step-by-step mechanism. Let's take a quick look at this clicker question. So this was saying you add a drug that inhibits DNA ligase. And if you remember, the answer here was E, because all the steps in the mechanism, which is shown on the right, can proceed until the part where you need DNA ligase. And so you end up with E, which is the DNA that's put together but not entirely ligated. But let's say instead of inhibiting ligase, you inhibit transposase. What product are you going to end up with in that case? A, right? So transposase, remember this little ladder thing here is the target DNA. Transposase cuts into the target. So if you've inhibited or shut off transposase, you can't even cut it to begin with, so you end up with what you started with. Take a look at B there. What would you need to do, what would you need to modify in order to end up with a product that looks like B? This is kind of a trickier question. Okay, so yeah, that is one. So if you're saying if you somehow modified this insertion sequence, you can prevent it from going in. That's a good answer. What I was looking for was you'd need to induce some kind of point mutation in transposase such that it was able to cut the target DNA but not insert the insertion sequence into the target DNA. So this couldn't be done just by like cutting out a protein entirely or shutting it off or inhibiting it. You would need to specifically modify the function of transposase such that it could do one of its functions, but not the other one. Any questions on that? So again, this is cut and paste, not copy and paste. So transposons don't actually replicate themselves unless this happens to occur where after the transposon is replicated, it moves in front of DNA polymerase and then gets replicated again. So this is an example of a retrotransposon, which is not cut and paste, it's the copy and paste mechanism. So this encodes two different proteins, not just one, which is a reverse transcriptase that makes a DNA from the RNA transcript of the transposon, and an integrase that then reintegrates it into the genome somewhere. So this gene just gets transcribed by RNA polymerase. We're going to talk more about transcription later in the course. But then the gene, the protein that this gene codes for actually makes this RNA back into DNA, and then the other protein that the gene encodes for inserts it somewhere else in the genome. So these actually get copied and pasted throughout the genome. So these are two basic classes of retrotransposons, lines and sines, difference being L and S for long and short. Long interspersed elements, lines, encode the genes that actually do this mechanism. Whereas sines are much shorter. They don't code for a gene. They just happen to have the sequence that is recognized by the reverse transcriptase protein. And so sines essentially take advantage of the action of lines without needing to encode all that information for the gene. So it's kind of like genomic ecology going on here, where lines are transposons that are already genomic parasites, but then you have sines that are parasites on the parasites taking advantage of the parasites' mechanisms. So it's interesting stuff. ALU elements are just an example of sines. I'm not going to read these bullet points here, but you can go through the slides. So just like organisms, these genomic parasites evolve over time. The sequences change over time. And so you can look at the sequences of signs of ALU elements, for example, and use them as a kind of tracer of phylogeny in order to help assemble evolutionary trees. So who can answer this question in red on the slide here? Why would you want to control transposons and keep them from just moving all over the place in the genome and replicating all the time? Yeah? They Yeah, so that's one good answer is if they're jumping all over the place, maybe they jump into the middle of an essential gene, and if that happens, for example, early in development, and it's a cell that is going to turn into a bunch of other cells, the organism's going to die. Another potential answer is, well, if transposons are just jumping all over the place and copying and pasting all the time, then eventually the genome is going to be nothing but transposable elements and none of the good stuff, basically. So all these resources, all of this energy, it takes energy to copy and paste genes and move them around. All this energy is going to be wasted on essentially a parasitic activity and not on essential functions, and the organism is going to die. So although transposons can have benefits, which are right here, they can also cause problems, and that's why transposons are largely kept silent through chromatin compaction, and RNA interference, which just kind of prevents the transposon genes from being expressed. Three quick ways here in which transposons can be beneficial is they can help kind of shuffle genes around and create variation upon which natural selection can act. So you can have gene duplication as a result of transposable elements. And then so you have two copies of a gene, and mutation can act on one of them and kind of experiment with it. But if you break that gene and say it's an essential gene, it's okay because you have another copy. So that organism's not going to die. So you can kind of experiment with this gene, but still have a working copy. Exon shuffling is another example. So it's maybe more for like a structural biochemistry class, but you can have modules of protein that get moved around based upon how exons are cut and paste to each other. So essentially shuffling the gene as an mRNA is processed. And by moving the exons around within the genome, you can create new proteins, moving the modules of the protein around. And then it can also, so shuffling genome around can also influence regulation of gene expression. Again, looking at this chart, 25% of the genome, we don't know what's up with that. It used to be called junk DNA. Why might that not be accurate? It might actually have a function. This is 25% of the genome, right? What are the odds that 25% of the human genome is completely useless? Say there's a mutant human that has that 25% useless DNA cut out, and now has a 25% smaller genome to maintain, and has to spend 25% less energy maintaining that genome. That mutant human would have a huge selective advantage over the rest of us, right? So for all the billions of years that we've been evolving from single-celled organisms, what are the odds that 25% of our genome is completely useless stuff? No, it would have been selected against. It's got to have some kind of function, whether it's coding, whether it's beneficial to just space genes apart, whether it's structural, something like that. It's got to have some kind of function, and we probably just haven't found it yet. So calling it junk DNA is questionable at best. Okay, last part. Out of these two things, we only covered gene cloning in class, so don't worry about inactivation, okay? Again, these are known as like forward genetics and reverse genetics. So in forward genetics, we're isolating a gene and studying that gene's function alone. In reverse genetics, you knock out a gene, you take it away, you shut it off somehow, and see what happens. We shut off this gene, and the mouse was born with no legs. That gene must have had something to do with like leg development. But in forward genetics, you take that gene out and look at it in isolation. Both of these techniques, neither one of them is perfect. They both have issues. If you just take a protein out, you take one gene out, you express that protein in bacteria, and you want to study its function, what could possibly go wrong? Exactly. You don't have the full system in place. All the other proteins it might interact with, all the things it might do in a eukaryotic cell, for example. That's probably the best answer. Another answer is eukaryotic genes expressed in bacteria often don't fold correctly, or they don't get expressed correctly. So you really need both of these techniques in combination to get the full concept of how a gene functions. Okay, this is just the basic model for how you isolate a gene and study it. We use restriction enzymes that cut very specific known sequences. They are endonucleases because they cut from within a DNA molecule rather than coming in from the outsides. And they create often these sticky ends that can be used to religate into something else. And so let's say you found your gene that you want to study, and you need to know what restriction enzyme can I use to cut that gene out. Well, what you might do is, assuming you have a sequence for this part of the genome, here's the genome, here's your gene of interest, you just kind of scan along here looking for a sequence that matches a certain restriction enzyme sequence. So, oh, I found one, this will work. And oh, here's that same sequence over here, this will work. And maybe these are in the middle of a neighboring gene, maybe they're not. But if they are, then you're just going to cut into the middle of that neighboring gene, and it's not going to be a big deal. And usually you'll have a vector. There's a range of vectors that have been designed, developed, that have the specific restriction enzyme sequences in the right place for you to ligate in your gene of interest. So you cut it out using restriction enzymes, you cut the vector, you mix the two things together, and you pour in some DNA ligase that combines them, and you get your vector with your gene of interest. And if your vector has a selectable marker on it, selectable marker is just a fancy jargon term meaning some gene on the vector that can be used to select cells that have taken up the plasmid. And often this is, it doesn't have to be, it's often antibiotic resistant gene. So because not all of the E. coli cells are going to take up the DNA necessarily. E. coli do not readily take up DNA. You have to do all sorts of tricks, shock them with electricity, just apply stress to them somehow so they want to go out and grab DNA. Only a few out of like your million cells in your culture, maybe 100 of them will take up the plasmid, and you only want to look at those cells that have taken up the plasmid. So instead of going through and looking at each cell and sequencing its DNA to find out which ones took up the plasmid, you can just select for it using media with that antibiotic in it. Okay. So the answer to this question was an origin of replication to allow the plasmid to replicate. But why might you want answer C in there, a restriction site? Yeah, exactly. So what I was just mentioning, where you want to be able to cut that vector open in order to insert your gene of interest. And a restriction site is one way to cut it at a very specific known location. Yada, yada, yada. Did I just skip through? Oh, I thought we got down to... We didn't get down there? Okay. Sorry. Okay, so that's all. Let's take a 5-minute break, stretch break. I'll go get some water. Everyone who feels good can leave. Everyone who wants to ask questions or go over things more in detail can stick around, and I'll be here.
I think a good idea might be, like we can all just stand by the board, and if people have questions, they can just come down and we can just answer individual questions. And then if you guys have individual questions, you can come down and ask us, and we'll draw it on the board.